Good afternoon, all of you. In the previous lecture, we have discussed about the journal introduction of the photosynthesis. Today, we will discuss about the steps that are involved in the photosynthesis and also the component or structure that take part during the process of photosynthesis. I hope you will enjoy it. Now, let's move on to the topic. In the earlier times, the photosynthetic reaction was written as, we can here write as 6 times carbon dioxide plus 6 times water in the presence of sunlight and chlorophyll. They manufactured their own food in the presence of sunlight and chlorophyll. That was C6H12O. So this is the glucose molecule plus 6 times oxygen. Okay. This equation, this photosynthetic equation, has put many plant physiologists in great confusion. It was presumed that the 12 atoms of hydrogen and oxygen comes from the water molecule, whereas this carbon 6 carbon atom in the glucose molecule, it comes from the main carbon dioxide. In this equation, it was found that the oxygen which is released during the process of photosynthesis comes from the carbon dioxide. As we all know that it is not true. This equation is not true. Many scientific evidences have shown that this oxygen which is released during the process of photosynthesis comes from the water and not from the carbon dioxide. So this equation is a wrong equation. Okay. It was a scientist known as von Neil who in 1931 performed an experiment using photoautotropic bacteria. Okay, this the photoautotropic bacteria is those bacteria which synthesize its own food using carbon dioxide and hydrogen sulfide. He uses a bacteria, photoautotropic bacteria known as Chromatium vinosum. This bacteria synthesizes its own food using carbon dioxide and hydrogen sulfide in presence of sunlight and special type of chlorophyll that is known as bacteriochlorophyll, or most probably chlorobium chlorophyll. This chlorophyll is present in the thylakoid membrane of the bacteria that is scattered in the cell of the bacteria, in the cytoplasm of the bacteria. These bacteria does not have the chloroplast. That is the organelle that perform the photosynthesis. They manufacture their own food, that is C6H12O6. They synthesize the sugar molecule plus they release the 6 molecules of water and also a byproduct, that is your sulfur. It is clear from the equation that von Neil has given that this sulfur is the byproduct. It is released from the breakdown of hydrogen sulfide. Here, carbon dioxide is reduced into sugar molecule. It is reduced into the carbohydrate. The energy of the reduction of this carbon dioxide into the carbohydrate, this electron and proton are provided by this hydrogen sulfide. Okay, here it is clear from the equation that if carbon dioxide was the source of the oxygen in the photosynthesis, then the byproduct was also the oxygen. But it is not the case. Here the oxygen in the carbon dioxide is used for the manufacture of sugar and hence it is not released. So Von Neal used this equation to give the model of the photosynthesis in the higher plant. He writes the equation for the higher plant, photosynthesis in the higher plant, as he replaces the hydrogen sulfide with water molecule. As we know that in the higher plants, water is utilized during the process of photosynthesis. Same with the help of sunlight and chlorophyll pigment. And also it produces the food, that is your sugar molecule plus 6 times water plus 6 times oxygen molecule. From this model of the Von Neal, it is clear that oxygen which is released during the process of photosynthesis comes from the water and not from the carbon dioxide. And also this, in the earlier equation, there were only 6 molecules of water. However, in this modern equation of the photosynthesis, here it is 12 molecules of water. The water provides necessary electrons and protons for the carbon reduction of the carbon dioxide to carbohydrate. That is, necessary electrons and protons are provided by this water to carbon dioxide to reduce it into the carbohydrate. Now there arises a question, there is a fact that how it is possible that water provides the oxygen, or water liberates the oxygen into the atmosphere during the process of photosynthesis. Many scientists have proved this using various models, using the various experimental evidences. One evidence was that of Robert Hill. He performed an experiment and gave it its name as Hill's reaction. He uses the green plant, that is Citilleria media. They synthesize the organic food using carbon dioxide and water, and also liberates the oxygen, but he uses the oxidizing agent as ferric chloride or DCIP. In another experiment which clear-cut give the evidence that oxygen which is released during the process of photosynthesis comes from the water. There were 3 scientists who have just proved that. They were 3 scientists. One was your Rubin, another was your Hasid, and Kamen, who in 1941 performed an experiment with a green algae, or you can say it as chlorophyce. They took the green algae, that is your chlorella, for the experiment. Okay, the chlorella is the unicellular algae, unicellular green algae. It is spherical and non-motile. Okay, it has the bell shape of chloroplast with the central nucleus without any flagella. It is the chloroplast pigment, it is the protoplast. Cytoplasm and nucleus. Both cytoplasm and nucleus we together called as the protoplast. They perform the experiment in the chlorella. During the process of photosynthesis, they use the carbon dioxide, and the water which they use has radio-labeled oxygen. Okay, has radio-labeled oxygen. The oxygen, usually the oxygen atomic mass is 16. Normal isotope of the oxygen is 16. The radio-labeled isotope of oxygen is O18. So they use this isotope of oxygen in water. During the process of the photosynthesis, what happens? It is 6 times carbon dioxide plus 12 times water. It forms the carbohydrate, that is C6H12O in the presence of sunlight and chlorophyll pigment. And also here the water is released as byproduct. The oxygen which is released during the process of photosynthesis, where they use the radio labeled isotope, here the oxygen which is released was radioactive. Okay, it is clear from the experiment that the oxygen which is released during the process of photosynthesis was radio labeled, same with that of the water that contains that this radio labeled oxygen. It is clear from this experiment that the oxygen which is released during the photosynthesis comes from the water and not from the carbon dioxide. They also perform the same experiment using the carbon dioxide radio labeled oxygen in carbon dioxide plus 12 times water. Here they found that the oxygen which is fixed in the glucose contains same radio labeled oxygen plus water plus 6 molecules of oxygen. From this equation it is clear that the oxygen is released during the process of photosynthesis come from the water and not from the carbon dioxide. Now we will discuss the side where the photosynthesis in the plants takes place. Now there occurs a specialized type of the cell which are found in all the green parts of the plant body. This specialized type of the cell is known as chlorenchymatous cell. Okay, this chlorenchymatous cell is a type of parenchymatous cell, that is they have thin wall and are living. Okay, this chlorenchymatous cell is present in all the green portion of plant body, that is they may are present in the leaves, they are present in the herbaceous stem. When this chlorenchymatous cell are present in the leaves, these are known as mesophyll tissue or cell. This is the mesophyll tissue or cell in the leaves. Inside the mesophyll cell, there occurs a specialized type of the plastid that is known as your chloroplast. Okay, this is a specialized type of the plastid found in the chlorenchymatous cell, that is all the green portion of the plant body. Okay, this chloroplast, when we see in the leaves, its number is about 500,000 per square millimeter. In 1 millimeter there is about 500,000 chloroplast organelles. Okay, now we have diagrammatically represent the internal structure of the leaf in order to understand the process of photosynthesis, the place where the photosynthesis takes place. So in the internal structure of the leaf, here the leaf is bounded by upper as well as lower epidermis, that is they are protective in function. Okay, the epidermis is followed by the columnar shaped cell with no intercellular spaces in between them. This columnar cell is known as their palisade parenchyma. Okay, this palisade parenchyma is followed by another loosely arranged cell with large intercellular spaces which are meant for gaseous exchange and also in the transportation of the water vapor. This irregularly shaped tissue is known as irregularly shaped cell is known as your spongy parenchyma. Together, palisade parenchyma as well as spongy parenchyma are together called as mesophyll tissue. Okay, inside the mesophyll cell or palisade or spongy parenchyma, the chloroplast occurs in the periphery of the cytoplasm. We can here draw the mesophyll cell, here the chloroplast occur in the periphery of the cytoplasm. Okay. The stomata, that is the pores which are regulated. The stomata occurs both in upper as well as lower epidermis, but the maximum number of the stomata occur in the lower epidermis. The main function of the stomata is that it regulates the exchange of gases, that is carbon dioxide and water, between atmosphere and mesophyll cell. So during the process of photosynthesis, carbon dioxide enters in the mesophyll cell through the stomata. Okay, and oxygen gets released from the stomata. Now, this cell organelle, that is your chloroplast, the structure of the chloroplast. In algae, there occur much variation in the shape and size of the chloroplast. We can get the different shapes of the chloroplast in algal group. For example, in Spirogyra, the chloroplast is spiral in shape, whereas in Zygnema it is star shape. In Oedogonium it is reticulate. In Eulothrix it is collar shape. In Chlorella it is bell shape. Whereas in higher plant, the chloroplast organelle is ellipsoidal or disc shape or biconvex shape. As you have clearly seen that it is biconvex in shape. It has a diameter of 4 to 10 micrometer. Okay, it has diameter of 4 to 10 micrometer and the thickness of 2 to 3 micrometer is. Okay, the chloroplast is in the higher plants, it is bonded by double membrane. Okay, the outer membrane as well as inner membrane. Between the outer membrane and inner membrane, there occurs a space that is known as periplastid space. This membrane, double membrane, is semi-permeable. That is, it regulates the flow of the metabolite between the cytoplasm and inside the chloroplast. The inner membrane is bond the thick hyaline granular matrix or liquid matrix that is known as your stroma. Okay, the inner membrane, it bonds the thick hyaline granular or proteinaceous matrix that is known as your stroma. Okay, the stroma is the site where the dark reaction takes place. You should note that the stroma is the site where the dark reaction takes place. The stroma contains water, it contains inorganic molecule, it contains coenzyme and all the necessary enzymes which are meant for the photosynthesis, for the synthesis of the food material. Okay, as we all know that the chloroplast is a semi-autonomous organelle because it contains its own genetic material, that is its DNA, and also it contains its own protein synthesis machinery. Here the genetic material is double-stranded circular DNA, just like that of the prokaryotes. Okay, and also the ribosome are 70S types. Okay, inside the stroma there occur in this stroma there occur a flattened sac-like structure that is known as your thylakoids. Thylakoid is a sac-like or vesicular-like membrane bond structure. Okay, these thylakoids get arranged one above the other like the piles of coin to form a structure known as your grana. Okay, these are thylakoids one above the other. These are known as your grana. Individual unit is known as your granum. There are about 40 to 60 grana per chloroplast. Okay, 40 to 60 grana are present in the chloroplast. These grana are interconnected with the other. These granum are interconnected with the other grana with the help of membrane lamellae, known as your stroma lamellae. Known as your stroma lamellae. Okay, it's known as your stroma lamellae, or it is known as your frat channel. Frat channel. Okay, the outer membrane of the outside of the thylakoid is in contact with the stroma, whereas inside of the thylakoid it is called as your loculus. Okay, or thylakoid space. The thylakoid space between the adjacent thylakoid in the granum remains continuous, or it is known as interthylakoid lumen. Interthylakoid lumen. Okay, this remains interconnected with the all other thylakoids in the granum. This granum is interconnected with the all other this neighboring granum. All the photosynthetic pigments are present on the thylakoid membrane. The thylakoid membranes are responsible for light reaction of photosynthesis, or responsible for light reaction of photosynthesis. Light reaction of photosynthesis, whereas stroma is responsible for dark reaction, or you can say that carbon dioxide fixation takes place in the stroma region of this chloroplast. The thylakoids contains all the pigments which are involved in the photochemical reaction. We can here see that when we see the location of the pigments in the thylakoid membrane, we can here see that the location of pigments. Location of pigment की पिगमेंट कहां पर लोकेट होते हैं? थाइलाकोइड्स पे. जो थाइलाकोइड्स में मेम्ब्रेन जो होती है, जो पिगमेंट्स होते हैं, as you know that the thylakoid membrane is just like the semi permeable membrane, or we can say that it is just like that of plasma membrane. इसका स्ट्रक्चर जो होता है वो सेम होता है as compared to that of the plasma membrane. There occur protein complexes in the thylakoid membrane. These protein complex जो होते हैं यहाँ पर, they get interacted with the pigment. जो पिगमेंट मॉलिक्यूल्स होते हैं, जो क्लोरोफिल मॉलिक्यूल्स होते हैं या कैरोटीनॉइड मॉलिक्यूल्स होते हैं, इनके साथ जो है ये प्रोटीन क्या काम करते हैं? नॉन कोवेलेंटली जो है अटैच रहते हैं टू फॉर्म पिगमेंट प्रोटीन कॉम्प्लेक्स. टू फॉर्म पिगमेंट प्रोटीन कॉम्प्लेक्स. देयर इन द थाइलाकोइड देयर आर अबाउट 4 टाइप्स ऑफ द कॉम्प्लेक्स. कितने टाइप्स ऑफ कॉम्प्लेक्स होते हैं? 4 टाइप्स ऑफ कॉम्प्लेक्सेस होते हैं. कहां पर? इस thylakoid membrane के ऊपर. First one, it is known as pigment system first. Second, it is known as pigment system second, or photosystem second. Third is known as cytochrome B6 F5 complex. Fifth one is known as your fourth is known as your ATPase complex. Okay, this pigment system first contains the necessary chlorophyll molecule and also some carotenoids and xanthophyll molecule. Okay, and form the light harvesting system first. Okay, This pigment system first is present in the non-oppressed part of the thylakoid membrane. That is, ये ऊपर की तरफ प्रेजेंट होगा, यहाँ ऐसे इस पोर्शन में प्रेजेंट होता है ये. ओके, दिस इज प्रेजेंट इन दिस इज द मेम्ब्रेन स्ट्रक्चर. आई हैव जस्ट थाइलाकोइड मेम्ब्रेन का मैंने इंटरनल स्ट्रक्चर यहाँ पर बनाया है. यहाँ क्लियरली आप यहाँ देख सकते हैं कि जो फोटो सिस्टम फर्स्ट है, इट इज प्रेजेंट ऑन द non-oppressed region of the thylakoid membrane, as well as this ATP इस complex जो होता है, जहां से आपके पास जो है ATP बनता है during the light reaction, is also present in the non-oppressed region. Okay, whereas जो आपके पास फोटो सिस्टम सेकंड जो है वो oppressed region में प्रेजेंट होता है. दैट इज, इसके यहाँ पर ये पिगमेंट सिस्टम सेकंड प्रेजेंट होता है. वी विल डिस्कस इन डिटेल इन द लेटर पार्ट ऑफ योर लेक्चर कि पिगमेंट सिस्टम सेकंड जो होता है वो कहाँ पर जो है वो लोकेट होता है, इसमें क्या कौन कौन सी कंपोजिशन होती है, ये फंक्शन कैसे करता है. ठीक है, जब हम लाइट रिएक्शन पढ़ेंगे, जब हम इसका ओवरऑल डिटेल पढ़ेंगे तो वहां पर हम इसको अंडरस्टैंड करेंगे. Second thing, third one is your cytochrome B6F5 complex. It act as bridge between photosystem second and photosystem first. जो नॉन अप्रेस रीजन में जो होते हैं आपके पास क्या प्रेजेंट होते हैं फोटोसिस्टम फर्स्ट, और अप्रेस रीजन में आपके पास प्रेजेंट होते हैं पिगमेंट सिस्टम सेकंड. ओके, in addition to these four pigment system, in addition to these four these complexes, there occur accessory mobile elements, that is your plastoquinone, and another one is plastocyanin. Okay, plastocyanin. जो प्लास्टोक्यूनिन होता है, इट इज अ मोबाइल इलेक्ट्रॉन कैरियर. दैट इज मोबाइल इलेक्ट्रॉन कैरियर मीन्स कि ये आपके पास यहां से मूव होकर आगे की तरफ या मेम्ब्रेन के ऊपर या मेम्ब्रेन के नीचे चलता है. ठीक है, वेयर एज प्लास्टोसाइनिन सेम, इट इज ऑल्सो दिस मेम्ब्रेन मोबाइल कैरियर, दैट इज इलेक्ट्रॉन कैरियर, जो आपके पास फोटो सिस्टम सेकंड से इलेक्ट्रॉन लेकर आपके पास आगे की तरफ इलेक्ट्रॉन ट्रांसपोर्ट चेन की तरफ बढ़ता है. ये आपके पास 6 कॉम्प्लेक्सेस होते हैं या 6 आपके पास चीजें होती हैं जो आपके पास थाइलाकोइड मेम्ब्रेन के ऊपर प्रेजेंट होती हैं. नाउ यू कैन जस्ट, देयर इज वन मोर एक्सेप्शन ऑफ पिगमेंट सिस्टम, दैट जो आपके पास क्लोरो और ब्लू ग्रीन एल्गी आपके पास है, या इसको बोलते हैं साइनोफाइसी, और अदर बैक्टीरिया, ये भी आपके पास फोटोसिंथेसिस परफॉर्म करते हैं, एज वी हैव जस्ट सीन इन द अर्लियर. ये आपके पास फोटोसिंथेसिस परफॉर्म करते हैं बट डिड नॉट इवॉल्व द ऑक्सीजन, एज आई हैव सीन द आई हैव जस्ट टोल्ड यू इन द वॉनिल केस. तो साइनोबैक्टीरिया और जो दूसरे बैक्टीरिया हैं उनमें जो होता है क्लोरोप्लास्ट जो है ऑर्गली नहीं होता है. इन देम द थाइलाकोइड जो होता है वो थाइलाकोइड्स में क्या होते हैं, ये पिगमेंट्स होते हैं. ठीक है, दैट इज क्लोरोफिल पिगमेंट्स आर डायरेक्टली प्रेजेंट इन द थाइलाकोइड्स. तो जो थाइलाकोइड्स होते हैं दे आर स्कैटर्ड इन द साइटोप्लाज्म ऑफ द साइनोबैक्टीरिया और फोटोसिंथेटिक बैक्टीरिया. वो स्कैटर्ड जो थाइलाकोइड्स होते हैं, जो साइक्लाइड स्ट्रक्चर जो मैंने यहाँ पर बनाया, तो वो वहां पर डायरेक्टली साइटोप्लाज्म में स्ट्रक्चर होते हैं. नेक्स्ट वी विल जस्ट स्टार्ट अबाउट द पिगमेंट सिस्टम इन द प्लांट्स. की पिगमेंट सिस्टम कौन कौन से होते हैं, क्लोरोफिल मॉलिक्यूल, उसका स्ट्रक्चर क्या है, एंड देन कैरोटीनॉइड्स. एंड आई होप यू हैव जस्ट एंजॉयड इट. इफ देयर इज एनी क्वेश्चन प्लीज कमेंट ऑन below.